to introduce Dr. Jonathan Glass, who's going to talk to us about new approaches to biomarkers and finding therapeutic targets. He's professor of neurology and pathology at Emory University School of Medicine. He's head of the neuromuscular division there, and he's director of the Emory ALS Clinic. He's best known for his research on the pathogenesis and prevention of nerve degeneration in neurological diseases, and for his work in human and experimental neuropathology, focused specifically on ALS. He's also led the recent first-in-human clinical trial of spinal cord stem cell transplantation in ALS, and his leadership in promoting new efforts in ALS therapeutics has led to his current position as my co-chair at the Northeast ALS Consortium, and it's truly been a privilege to work with John for the last 3 years. John? Come on up, Dr. Glass.
Thank you. Thank you, Terry, for that very generous introduction. I'm very happy to be here. And I'm honored to be able to give this presentation. I gave this presentation last year as well on a different topic. Today I'm going to talk about something different. And what I'd like to do is to present to you how we get to where we are. So for many of you, this is extraordinarily important. How do we get to the kinds of drugs or therapeutics that we think might work for this disease? And there's a lot of basic science behind that. And what I'm going to do today is present some of that basic science. And I apologize to those of you who were here this morning and heard my presentation because a portion of it is going to be the same. The first part will be the same, about frontotemporal dementia. And the second part is about frontotemporal dementia, but it's brand new data that literally has just been published in the last few days. But it shows, and it looks like really, really complicated science, and it is. But if I can't explain it to you, then it means I don't understand it either. So it's really important. How do we find new targets? How do we design new drugs? And how do we get to the idea of the kind of clinical trials that James Berry is going to talk to you about after this? So without further ado, I'm going to talk about ALS and frontotemporal dementia. Frontotemporal dementia has really come into the limelight in ALS clinics. It's something that we have rediscovered because this is not new. It's been around since physicians and scientists have looked at ALS for more than 100 years. But it is a very prominent problem. It is a disease that makes a bad disease even worse. All you need to do is to speak to family members who have a family member with both ALS and frontotemporal dementia. And there's a lot of overlap between ALS and FTD. And I'm going to show you a little bit about that and where we're moving toward in terms of understanding targets for this and also designing clinical trials. There are many types of frontotemporal dementia. The one that's seen in patients with ALS is called the behavioral variant. These patients become disinhibited. They sometimes exhibit childlike behavior, inappropriate behavior, have a lot of difficulty in social situations. They sometimes become angry, paranoid. They do repetitive types of things, and it is extraordinarily disruptive to a family to have this disease, and then put on top of that the dysfunction that comes along with ALS, and it really is a problem. But there are other types of frontotemporal dementia that we typically don't see in patients with ALS. There's something called primary progressive aphasia, where patients have difficulty actually speaking. They know the language, but they can't get it out. And then something called semantic dementia, where patients make mistakes in the types of words that they use. It's argued that about 20%, probably 25% of patients with ALS may develop dementia, but in fact a larger proportion of them might develop what we call cognitive impairment, where you wouldn't necessarily recognize that these folks have a a dementing or a cognitive type of illness, but if you do formal cognitive testing on these folks, they may have some abnormalities that you don't expect. What we don't understand is whether those folks with the minor cognitive abnormalities end up having true full-blown dementia, as is typically seen in patients with Alzheimer's disease. So this is a video. If you could play this for me. This is a patient that I saw recently, just to give you an example if you haven't seen this before.
How you doing?
This is a man who has ALS.
Why are you here?
He is a high-powered lawyer in town who now is completely non-functional.
Tim, can you tell us what's going on? Okay, Liz. Come on back, Tim.
He's gonna punch that button.
This is pretty hard to watch. And
Okay, we're gonna go sit
trying
down
to help
again,
the
okay?
family has been quite difficult as well. We have patients, you can go to the next slide, or I can go to the next slide. We have patients who will get phone calls in the middle of the night, what do I do, anger outbursts, inappropriate behavior, things like that. So again, add that on top of ALS, and it's a huge problem. But in fact, this is a problem that's been known for a long time. Here's a paper from the Mayo Clinic in the 1930s titled Psychotic and Emotional Phenomenon in Patients with ALS. And if you look at this, you can see that these patients clearly had frontotemporal dementia. Things like paranoia, suspecting that her husband was with other women. She'd rarely forget things in the immediate environment, even if what she might be doing. She clearly has ALS. Her tongue is weak and atrophied. She has fibrillations, or what we call now fasciculations. You can see here she would act in a bizarre manner where she would dress in every opportunity, putting her clothing on over her night clothes. So again, this is back from the 1930s. This isn't anything that's new. Another case of a farmer, again from the 1920s. He had ALS, but also became irritable and quarrelsome, and in fact would stand around staring, doing nothing. So not a new phenomenon. And then the other thing that we have not, this is not new, is that the idea that this can run in families. And these are three first cousins from a group from 1973. You can see here, 1, 2, 3. Each of them had either ALS or FTD or both. And so there's a genetic component here as well that has been around, has been known about for many, many decades. And here are the three folks, a guy who's got ALS clear, and also the FTD, the irritability and hypersexuality, the disinhibition. Another person who's a first cousin who has ALS clearly, not clear whether this person has frontotemporal dementia. And then again, another cousin, inappropriate behavior. And so frontotemporal dementia, ALS, and frontotemporal dementia all in the same family. So the neuropathology is quite striking. When you see the brain of somebody with ALS and FTD, that's a normal up top, and this is what it looks like here. What you can see here is the frontal lobes are quite atrophied, as are the temporal lobes. The ventricles are quite big, and the brain is shrunken. We've seen brains that look almost like walnuts, and it's not absolutely clear why that happens. But this idea of the neuropathology of ALS and FTD overlapping, again, is not new. Neuropathologists have known for a long time that the neuropathology that they see in patients with ALS, they sometimes see similar things in patients who die with dementia. And so they called it motor neuron disease inclusion dementia. So it turns out that one of the early findings in patients with ALS was that in their neurons and some other cells, there are these inclusion bodies. It was found later on, typically in the 1980s, that these inclusion bodies were made up at least of a protein called ubiquitin, which had something to do with the breakdown of abnormal proteins. And in fact, this was found as well in patients with frontotemporal dementia. And in fact, it was brought up in 2005 that maybe ALS and FTD pathologically are connected. That's 2005. Then in 2006, this paper comes out, and this is a group from the University of Pennsylvania. This is extraordinary work that really changed the course of ALS research. They defined these inclusion bodies in patients with both ALS and frontotemporal dementia as containing this protein called TDP43. TDP43 is a protein that is important in shuttling DNA and RNA inside and outside of the nucleus and moving it back and forth. And these inclusions are present basically in all patients who die with ALS, probably except for the patients who have SOD1-related ALS, which is a relatively small population, about 2%. And about 50% of people who die with frontotemporal dementia also have these TDP43 inclusions. And so here's the connection pathologically. We've already shown you the connection clinically between ALS and FTD. I've shown you a connection genetically in the same families between ALS and FTD. Now I've shown you the connection pathologically between ALS and FTD. So these two diseases are linked. And so if we're going to study ALS and we're going to try to treat ALS, we have to think about the pathophysiology and the underlying biology of frontotemporal dementia as well. So let's get to the genetics. And genetics in ALS really is the forefront of research as far as I'm concerned. It turns out that there are many, many different genes that can have mutations that either cause this disease or drive the disease and increase the risk of getting the disease. And the first one, of course, was back in 1993, SOD1. And SOD1 was found in several families and, in fact, was quite an exciting discovery back then because we thought it was going to lead to understanding this disease and hopefully a cure for this disease. Unfortunately, it hasn't happened that way several decades later. But in fact, it created a large number of models of ALS in mice, rats, worms, flies, zebrafish. And so you can take human mutations of this SOD1 protein that cause ALS, introduce them into different animals, and these animals actually get a disease that looks like human ALS. And so it becomes a model for us to, number one, understand the biology of the disease, and number two, potentially to test new treatments for the disease. But SOD1 was just the tip of the iceberg. Over the years, many other genes have been discovered, and this is no longer up to date. There are several others that are not out here. And the size of the circles represent the percentage of families that actually have this mutation. And there's one that's particularly large, and that's this one called C9orf72. C9orf72 was described in 2011 by two groups, Rosa Rademakers and Brian Traynor. And it turns out that this gene mutation, which is a relatively unique gene mutation, accounts for about 40%, 40 to 50%, at least in this country, of families who have ALS. Now, even though that's the case, it's not 40% of ALS because all of these mutations combined are only about 10 to 12% of ALS because that's the percentage of ALS that runs in families. So there are some mutations that will cause frontotemporal dementia. Here, tau mutations and granulin mutations, other ones that will just cause ALS without dementia, including the SOD1 mutation. But there are some that can cause either or both. And in fact, in here is that C9orf72 mutation, which is I'm going to spend time showing you a little bit now. This is a relatively unique mutation. This is called a repeat expansion disease. We've known for a long time that there are repeat expansion diseases that cause different types of neurological diseases, things like Huntington's disease, Friedreich's ataxia, mental retardation, can all be caused by expansion mutations. And those are usually what we call trinucleotide repeats. Those are three nucleotides or three bases that have been expanded abnormally that cause the disease. quite interesting. But in fact, this is 6 nucleotides, or a hexanucleotide repeat disease. And the hexanucleotide that we have, and for those of you who've done a little bit of biology, DNA is ATGC. We have 4 types of bases. And so this is G and, this is G and C. And the G and C goes GGGGCC. GGGGCC, and it repeats itself. And all of us have repeats. Typically we have, those of us who don't have disease, about 2 to 10 repeats in our genome. But folks who have ALS or FTD with this repeat can have hundreds or thousands of these repeats, and that has complications. The hundreds or thousands of these repeats can actually cause abnormalities that probably cause stress on neurons and possibly cause disease itself. And this is the way we see these things. This is called repeat prime PCR. This is what a normal looks like, where there isn't a lot of repeats out here. Each one of these things represents one of those repeats. And you can see out here there are dozens and dozens of them, and eventually it goes out to hundreds or thousands of them. And so, at least in my clinic, we screen everybody for this because it is relatively common, and there is reports of patients who have what we call sporadic disease or no family history of disease that also may be carrying this mutation. So what does it do? So what this mutation can do is introduce a new kind of biology that at least I didn't know anything about when it first came out, and it's the idea that this DNA sequence can make protein without the typical machinery that it needs to make protein. So typically, when your DNA is coding for a protein, it actually has a sequence in it that says, start reading here. We call that a start sequence. Start reading here, and the DNA is read there to make what's called RNA, and the RNA is then read to make proteins, and that's what we're made of, is proteins. And so what this does, instead of having that start, what we call a start codon, it doesn't need a start codon. And in fact, the DNA machinery and the RNA machinery can read it without it. And when it does that, it can make these things called dipeptide repeat proteins. And so an amino acid, proteins are made up of amino acids, and each amino acid is coded for by 3 of those DNA bases. Okay? So if you have 6, you can have 2 amino acids, and we call that a dipeptide. Make sense? Okay. Stop me if I'm not making sense. I got to make you understand this. Okay? And so if you have GGGGCC, what that codes for is this GGG codes for the glycine, and this GCC codes for an alanine. And so that becomes a GA dipeptide protein. Okay? GA dipeptide protein. But if I decide to read starting on the next one, instead, I no longer have GGG-GCC. Now I'm going to read over here. And now I have GGG-CCG. And that's now a glycine and a proline, or GP. And that can go on, or GGCC, glycine arginine. And you get the point. Depending on where you start, you can get 2 different peptides. And in fact, to make it more complicated, you don't have to read it left to right. You can read it in Hebrew and go right to left. And you can get 2 different types of dipeptide proteins as well. And so there are 5 different dipeptide proteins that can be made from this. And if you think about it, I told you there are hundreds or thousands of these things. And so if you have hundreds or thousands of these things that are all making these dipeptide proteins, you now can form these things, which are inclusion bodies made of these dipeptide proteins in the neurons. And it turns out, at least in cell culture, these things are toxic to neurons. So is it possible that making these dipeptide proteins actually are what causes the disease and cause neurons to die? Remember that, because it's going to be important, okay? But there are other things that this can do. Remember I told you DNA has to go to RNA, right? So if you're making all this DNA to make dipeptide proteins, you have to make all this RNA to make dipeptide proteins, because DNA has to go to RNA before it makes a protein. And when you make all that RNA, you can actually, the RNA starts to congeal together and makes what are called RNA foci. And in fact, those have been shown to be potentially toxic to neurons as well. So there's another possibility of how this is happening. So there are 3 major ideas of how this works, okay? Number one is, or let's start with number two, that these RNA foci actually can suck up, or they become sticky and they suck up the other RNA in there, and it makes the cell sick because it doesn't have the RNA to make the proteins. Another possibility is that these dipeptide repeat proteins are actually toxic and they kill cells. But another idea, and again, try to remember this one because I'm going to show it to you later again, is that this repeat allele, we have two alleles for every gene that we have. Remember, we have two chromosomes for each, or two alleles for each chromosome. That the one that actually has the expansion doesn't make enough of the normal C9orf72 protein. Because the protein is made for something, it does something. And if you don't have enough, maybe that causes the disease as well. And there's some data out there arguing that people with this disease don't have enough of this protein. They don't make enough. So those are the three major ideas. And another major idea that is really coming into the limelight now is the idea that these different biologies can get in the way of transport of molecules inside and outside of the nucleus. For a cell to be healthy and for a cell to function the way it's supposed to, not just a neuron, but other cells as well, it has to take that DNA and move it out to RNA, make proteins out here, and things are going to move back and forth. Machinery has to move back and forth in order to make this whole thing work. And it turns out that there are these things called nucleopore proteins. There are hundreds of them that actually are important for transporting these things inside and outside. And these different RNA foci, or in fact the repeats, or the dipeptide proteins can actually get in the way of that. And that's another possibility of how this disease works. So now we have lots of targets. We have lots of targets, at least for this type of disease. So how are we going to use that biology, that basic science, to actually get to an idea that maybe we can, number one, understand what causes the disease, and number two, design a trial or design a treatment that actually might be able to work. So the group at the Mayo Clinic, led by Len Petrocelli, has developed this assay for that GP protein, this polypeptide that I showed you before. And in fact, in patients who are carrying that C9 protein, they can measure it in their spinal fluid. And that's going to be really important because one of the problems we've had in clinical trials over the decades is that we give drugs to people, but we don't know whether they actually hit the target that we were shooting at. We can do it in a mouse, we can do it in a cell culture plate because we can see things. But in a human, how do you know if it actually got there? We need things called biomarkers. We need things called target markers. And in fact, here's something that we can measure in the spinal fluid that's directly related to this specific biology that I've just described to you. And what you can see also is that in people who are asymptomatic, they're carrying the gene mutation, they're in the family, but they don't yet have disease. They also have, some of them at least, have elevated levels of these dipeptide proteins. So now we have a marker. We have a biomarker of disease, a biomarker of the biology, and potentially something that'll tell us whether we've hit our target or not. And how are we going to do that? Well, turns out that not only can you measure it in the spinal fluid, you can measure it in what are called peripheral blood mononuclear cells, just from blood, because it's easier to do a blood sample than it is to get spinal fluid. But also, you can see if you put these things into culture, they actually are excreted from those cells, and we can measure them in culture. And if you then take something called an antisense oligonucleotide, this is the new biology in terms of therapeutics. If you can take a short piece of DNA or RNA and that binds to that DNA or RNA and keeps it from making those proteins, maybe you can actually make a therapeutic. And so here are antisense oligonucleotides, and what you can see here, that as you treat, the amount of these GP proteins goes down. That's really important. But even more important, if you have a mouse model that actually carries this mutation and can make these dipeptide proteins, as you can see here in the brain of this mouse, if you treat it with this antisense oligonucleotide, they go away. Or at least they're prevented from being formed, okay? And so if these things are important in terms of toxicity, then maybe we have something that we can do something about. Okay, so what do we do? We actually look at humans and we say, okay, what happens to these things over time in people who have ALS or frontotemporal dementia? And the good news is this, they don't really change over time. And that's good news because if we then give an antisense oligonucleotide to these patients and the amount of this stuff goes down, it means we've hit our target. We've done the biological manipulation that we tried to do, the same thing that we did in the mouse. And that is unique. We have not yet been able to do that in humans. It doesn't mean it's going to work. It means that we've actually addressed the biology that we think that's important. And in fact, a company here in Boston called Biogen, along with a company in California called Ionis, who makes these antisense oligonucleotides, is designing a trial, and this will hopefully get started early in the spring in patients who are carrying this C9 mutation. And all you need to do is think about it. If it works, can we actually prevent disease, either in ALS or in frontotemporal dementia? Yes, this is not everybody with ALS, but it would be a huge win for the community if we could at least do this for this subpopulation of patients with ALS. So now I'm going to get to the harder core science that comes from my laboratory. The real question we had was, why? Why can patients with ALS get FTD, and why do patients with FTD get ALS? They're really different diseases. So ALS is a motor neuron disease. It shows up in a neuromuscular clinic. And frontotemporal dementia is a cognitive disease. It shows up in the dementia clinic. Those are really different. In fact, in our institution, there are two different sites. Those patients aren't even seen on the same hallway. And so how can they possibly, what's the biological basis for this? I've already told you there's pathological, genetic, and clinical overlap. But why is that happening? So we took an approach called proteomics. And these are the folks who I work with, a very talented graduate student in the laboratory, Imfan Umo, Nick Seyfried, who runs our proteomics laboratory, along with Eric Dammer. Great colleagues who are much smarter than I am, I just got to tell you that. And so here's the idea, that we're going to actually take autopsy brains from patients with ALS, patients with frontotemporal dementia, and patients with ALS and frontotemporal dementia, and then patients without a neurological disease, and we're going to grind them up, and we're going to throw them into this thing called a mass spectrometer, and we're going to do this thing called proteomics. And I'll describe that in a little bit. And we're going to see how it turns out. What's the difference among all these things? Completely unbiased. We're just going to dump them all in together and see if they actually come out to show us what the differences are. Now, we chose the frontal cortex. Why did we choose the frontal cortex? Because what I'm really interested in, all of these patients that I see anyway have ALS, but only a subset of them have frontotemporal dementia, and that's the frontal cortex. So I want to see what the difference is there versus people who just have ALS. And so what you're seeing here is kind of how we do it. We take these different groups, we put them through this thing called a mass spectrometry, and we get quantification of the different proteins, and we get thousands and thousands of proteins out of here, and then we do something called protein network analysis. And so why do we do this? Okay, why are we looking at proteomics? Well, because we're made up of proteins. Our whole brain is made up of proteins. Everything we have is made up of proteins, and so that's what makes us, okay? And proteins are the final product of the information that you inherit from your parents. So all of your DNA that you got from your parents is coding to make proteins, okay? So we're not just looking at the DNA. And in fact, we know that neurodegenerative diseases are proteinopathies. And I've already told you about the inclusion bodies in patients with ALS. Inclusion bodies and abnormal proteins are really proteins, and that's why we want to look at proteins. And so if you look at complexity, we start out with a genome, which may have maybe 25,000 genes, okay? But in fact, as you go up, those 25,000 genes make millions of proteins. And that's really what we want to look at. What's the difference there? And so if you're thinking about it, I'm going to get thousands and thousands of proteins. What am I going to do with that? I'm going to have a computer filled with all this data. how do I decide what's real and what's not real? How do I decide how to group them together? And we use something called weighted co-expression network analysis, or WGCNA. And what this does is it actually groups proteins together based on their similarities of how they change. And it's a completely unbiased approach. It's not that we say, take these proteins and see if they work together. It's take all the proteins, the thousands of proteins that you've measured, and see which ones kind of go up together, which ones go down together, which ones are associated with each other. We call that co-expression. And so we bin those proteins into what are called modules, and we can see by pattern recognition, and this is the kind of patterns that come out, which ones kind of group together, which groups of proteins are together, okay? And so this is what we did. Here we have 10 control grains, 19 patients with ALS, 10 patients with ALS and FTD, and 12 patients with FTD alone. And we do this analysis, and we make sure we take out what are called outliers, because we've got to be able to measure these proteins in all of these cases, otherwise it's not fair. And then we do something called differential expression, which is typically what we do. What's the difference between a control and a disease? Individual proteins, okay? But that's hard to do when you have thousands and thousands of proteins. And then we do this weighted correlation analysis, which is really the power behind this. And so we did it. And this is what it looks like. In this kind of diagram, what you can see here is ALS versus ALS FTD and just FTD. And this is just the differential expression analysis, just looking at proteins. And these are the unique proteins for ALS, the unique proteins for ALS FTD. And you can see in the frontal cortex, as you would expect, because these patients are demented, that there are many, many more proteins here that are unique to the dementeds, okay? But here's the real kicker, and I'm going to show you this one, and try to understand this. Again, we didn't tell the machine who had what. We just threw it all in together. It all goes in together. And the machine decided which ones are similar and which ones are not. And what it did was, and this is extraordinary for this kind of analysis, it actually separated them out based on their clinical features. The ALS patients were different than the FTD patients. The ALS FTD patients were somewhere in the middle, and the controls are also different. And this is without telling the machine. The machine actually made this decision, which tells us that there is a biological feature up there that actually is separating out these different cases. Not only that, make one more comment here, is that we actually had patients with the C9 mutation I told you about and the non-C9 mutation, and that actually didn't separate out. That wasn't a feature that actually separated these out. So the genetics didn't do it. It was just the clinical features. These patients have different biology when they come into the clinic. In fact, the argument is that they're really different diseases that are showing up together. And so what happens with this co-expression analysis, it takes these proteins and he bins them into different what are called modules. And we give each module a color. And we give it a color, and within that colored module are proteins that kind of fit together. And you can see here that some of them are related to RNA binding, or they may be due to inflammation or blood-brain barrier abnormalities. And these are, again, thousands of proteins that have been binned together in different groups of proteins that are what are called annotated, or this is the kind of protein it is. This is what we think it does, okay? And then we go back and we say, well, this is just data. Is it true? And we have to validate this. And so when we validate it, we say, well, what are these proteins in, and do they fit in the right cellular modules? And we found that the inflammatory proteins are in what are called the microglial and astrocytic cells, which are the cells of the nervous system that are involved in inflammation. We find that the synaptic proteins, which are involved in structures of neurons, are actually in the neuronal components, and the myelin proteins, which are important for conduction of electricity in the brain, the insulation around the different axons, form out in the oligodendrocytes, which make myelin, which tells us this makes sense. It's internally consistent. And then we can actually then start to compare. So we're going to compare the different groups in these different modules. And what you can see here is that some of them show that in the frontal cortex, the difference between control and ALS, as you would expect, is not a lot in these different modules, okay? Because the frontal cortex is not as affected in ALS as in frontotemporal dementia. But as you move to frontotemporal dementia with ALS or frontotemporal dementia alone, you start to see these major differences in these different protein groups. Okay, these are not individual proteins, but protein groups co-expressed. And what's really interesting here, and those of you out there who have seen a patient with ALS and frontotemporal dementia versus just a patient with frontotemporal, they're just as demented. There's no difference. It's not that the dementia is not as bad in an ALS patient. It is as bad as a patient who doesn't have ALS. But look at their protein expression. They're different. So an ALS FTD patient is different than a typical FTD patient, which may suggest to us that the frontotemporal dementia, even though it looks the same in these two patient populations, maybe there's a different biology underneath it. It's similar, but it's not the same. And then there are some things that are up in all diseases, and some of them actually go down. And these make sense, including the synaptic proteins that go down, because we're losing neurons in the frontal cortex. And so we decided, okay, this makes sense. Let's see if we can actually see this stuff. And what we do is actually, this is called, I call it the Ferris wheel, but it's actually called an igram for something called an eigenprotein. And these proteins in the middle are the ones that drive these different modules. These are the ones that are most strongly correlated. And if we go back and we look at some of these individual proteins in these modules, we can see that they're different the same way we saw them in the whole module, these individual proteins. These are astrocyte proteins, these are microglial proteins, and in this module you can see that they change the same way as the whole module does. And then we can go to the next step and say, okay, can we actually see this protein? And these things up top are called Western blots, and these are looking at specific proteins. And you can see here that in the FTD patient versus the ALS FTD versus the ALS, you can see that it goes up the same way it seemed to go up in those little bar graphs that I showed you before, and this is real protein. This is not the stuff coming out of the machine. This is actually taking the brain, grinding it up, and looking for the protein itself, and you can see that we can quantify that as well. And we can go one step further, and we can take these same brains, and then we can stain them with something called immunohistochemistry, which have antibodies that bind to these individual proteins, and you can see here that there are differences between the ALS and the ALS FTD, both in the upper portion, the cortex, and in the deeper portion in the white matter for all of these different proteins. And I think you can see the differences here and how much more staining there is in the FTDs. And then we can look at individual cells and really confirm that these are the cells that are making these proteins. Why am I telling you all this? It could get really boring. But why am I telling you all this? Why? Because now we have new targets. Now we have new proteins that we say, aha, this one seems to be different in this disease versus control, in this disease versus ALS versus FTD. But also now we have groups of proteins. And groups of proteins actually define pathways of biology. This is a pathway that may be important for causing ALS, for causing FTD, or for causing ALS and FTD. That's why this is important. This is how we get to the next step. Now we go, we make models, we see if we can create those models that actually look like these diseases, and then we can interact with these proteins by turning them off, turning them on, whatever we think is important in terms of treating the disease, and then again, we're at the next step. One other thing I was going to tell you is that one of the hypotheses was that there wasn't enough of this C9 protein. Well, when we went back and just looked at the ALS patients without FTD, and we just with the C9s, we actually found less protein, but that's something to be looked at in the future. So the summary here is that quantitative proteomic differences in the frontal cortex are not driven necessarily by the pathology or by the genetics. It's driven by the clinical features of the patient. So the clinic is still really, really important. And the C9 genotype, as I told you earlier, did not correlate with any of those co-expression modules, which again tells us, but maybe some of the biology that's occurring in C9 may be similar to to other types of ALS and FTD, and maybe the treatments that we have can overlap. We found 8 protein modules that were identified that differentiate these clinical phenotypes, and the inflammation module, which I showed you in the astrocytes and the microglia, was a consistent characteristic of the protein expression in the patients with ALS and FTD. And then we compared the C9 positives to the C9 negatives, getting back to that group, and only inflammation was important, and that even in patients with no dementia, had increased astrocytic and microglial proteins. I didn't actually show you that data, but it's important because it correlates the C9 patients with the other patients as well. And I probably went over, and I'm sorry, but that's how we get to stuff that we do clinical trials on, which James is going to tell you all about. Thank you.